So the research we are presenting is looking at our investigation into the impact of cold temperature on sickle cell disease pathophysiology using work that was previously done at the Rivers lab by studying the impact of abnormal retention of mitochondria in red cells. And so during the normal development of the red cells in the bone marrow, we expect them to lose their nucleus and then also the mitochondrial content. But the Rivers lab was the first lab to actually report that the sickled red cells abnormally retain mitochondria. And we believe that this plays a role in the downstream effects on the disease pathophysiology. And so this specific research looked at the impact of that and then also on cold temperature in mitochondrial clearance, in red cell clearance. Thank you. So we use the Towns sickle cell disease model. The Towns sickle cell disease model is a mouse model that has the human phenotype knocked in, and so it expresses the human hemoglobin. And so we used that, and then we used flow cytometry as a method to be able to stain the red cells. We to assess the red cell lifespan, we used the interaction between biotin and streptavidin, and then tracked the red cells over time, whilst also staining for mitochondria and then other markers. And then based off that, we're able to analyze the data and extrapolate and know the red cell clearance over time. But we're also able to perform other experiments where we exposed the mice after ethical clearance, of course, to various temperatures, and then assess these other parameters as well to see the impact of cold also on their physiological parameters. The key finding we found was that red cells that retain mitochondria have a higher clearance from circulation compared to those that don't retain mitochondria. And so we believe that mitochondrial retention may be playing a role in the longevity or the lifespan of the red cells. In addition to that, our cold experiments showed that exposing the mice to cold actually reduced the intracellular calcium. This is a very interesting phenomenon which we are interested in investigating to understand why and how it affects the disease pathophysiology. The mouse model may not fully recapitulate, the human pathophysiology, and so we are also performing. We have an air-cooked protocol in place, and we are performing some experiments using also human samples. So those are some of the limitations. Yes, I think the approval is exciting, but I also think that the research being done at the Rivers lab is the most exciting work happening in sickle cell disease right now. Investigating the role of these mitochondria and these organelles in the red cells is very important. And so I think that while the gene editing methods are exciting, it's very, very, I think what excites me the most is research that looks to create affordable solutions for underserved communities, because majority of the sickle cell patients are underserved. And so the research being done in the Rivers lab potentially in the future could be used by translational scientists to help develop small molecule drugs, but also other research that is going on, even currently at Berkeley, that is looking at creating affordable also gene editing solutions for majority of the sickle cell patients. I think that would be more impactful.